Welcome back to Gene Quest, where science meets fun. Today, we are diving into something straight out of a crime lab or a gene research center. It's DNA extraction. But guess what? We are not doing it with fancy lab equipment, but we are just doing it right here in my house with an onion. So if you're ready to get your inner senses on, let's jump in. What is DNA? So before we get our hands dirty, or rather soapy, let's talk about what DNA is. DNA, or is also known as deoxyribonucleic acid, is the blueprint of life. It holds instructions that tell cells how to grow, function, and even what color our eyes will be. So every living thing has DNA, including this onion. And now, today we are going to pull it out and see it with our own eyes. Exciting, right? Okay, scientists, here's what you need for our experiment.
Onion, 20 ml dish soap, 20 grams salt, hot water, 10 ml of cold ethyl alcohol, mortar and pestle, paper coffee filter, clear glass stick, 100 ml distilled water, and measuring spoons.
Got everything? Great! Let's get experimenting. So, what we are going to do first is have our onion bits. And we're going to put it right here in our mortar and pestle. So you can see here we have the onion bits into the mortar and pestle. So what we're going to do next is smash it gently. Why do we need to smash it? Because we need to break and open these cells. Now I know onions make you cry, but science is worth a few tears, right? After smashing, we need to put this onion, or should I call smashed onion, in a separate glass. With, of course, the use of a spoon or a spatula. So here's your smashed onion bits. For the next step, we will be needing a glass, and we will be going to mix here the 20 ml dishwashing liquid, 100 ml distilled water, and of course our 20 grams salt. And we are going to use measuring spoon for us to measure it accurately. The next step, we are going to pour this mixture in the smashed onion. And stir gently. This helps breaks the cell membrane and releases the DNA. Next step is exposed to heat. So in my case, I used a hot water and placed the mixture over it to warm it up. Now, since we are done exposing this mixture to heat, we are going to wait and let it cool down for 5 to 10 minutes. Next up, we are going to use a coffee filter because we are going to separate this liquid from the onion bits, because we only need the liquid because that is where the DNA is hiding. Since we are done separating the liquid from the onion bits, as you can see here, it's only liquid. We are going to use an ethyl alcohol, a cold ethyl alcohol, and pour it here in our empty glass. And afterwards, we are going also to pour this one here in the empty glass. So I forgot to mention, we also need to put the ethyl alcohol in these over these ice cubes so that it will become cold. Now for the magic moment. Slowly pour in a small amount of the mixture to this cold ethyl alcohol. So we will wait for it for about 5 minutes, and that's already.
5 minutes later.
See those white stringy clumps forming? That, my friends, is pure onion DNA. How cool is that? So how did we do this? Well, the dishwashing liquid right here broke open the onion cells, releasing the DNA. The salt helped the DNA strands to stick together. The alcohol, the DNA does not dissolve in alcohol, so it clumped together, making it more visible. Boom! Science at work. And there you have it. If you had fun extracting DNA today, don't forget to like, subscribe, and drop a comment down below. Thanks for tuning in, future scientists. Keep asking questions, keep experimenting, and I'll see you on the next one. Bye!